So first of all, thanks very much for inviting me. And as you've noticed, I've actually changed the title. So I went for Building Better Transcriptomic Maps to Build Better Therapies. And so I was going to start by asking why we bother with maps at all. So this was an excuse to be able to put an Indiana Jones and the Raiders of the Lost Ark picture up. I realized actually on an audience that I put this on before that was maybe on the younger end that they didn't necessarily know Indiana Jones, so it didn't mean very much to them. But hopefully this audience does know Indiana Jones. So why do we have maps? We have maps to navigate to new places. We have maps in order to be able to understand the relationship between geographical features. We have maps to plan and make decisions. We have maps to find treasure, most importantly, as in Indiana Jones. So why have a transcriptomic map, and what is a transcriptomic map? Well, a transcriptomic map, quite simply, is a map that actually relates to our genome map, which tells us where the exons and introns are, what the RNA that's generated from those genes actually is, and what the possible transcripts that might be coming from a locus are. And actually, the transcriptome map is much more varied and much more diverse than the genomic map is. So I didn't put up the number of genes according to annotation. This is gen code data. But actually, in fact, it wouldn't be the hundreds of thousands. It would be the tens of thousands you'd have on the y-axis for genes. Whereas on transcripts, you have this huge world of potential transcripts. And actually, to make advances in genomic medicine, we don't just need an accurate map of genes. We need an accurate map of transcripts. And that map is only second in importance to the genomic map itself, and it's actually still being updated. So as you're seeing on that plot, these are annotations going from 2003 to 2024, and they are changing. So why do I need a good transcriptomic map? Why would I want one? So I'm a clinician as well as a scientist, but I want to actually have that map because actually if I'm in a clinical situation, what I'm actually doing is seeing patients in clinic and asking what genetic variants have actually generated that phenotype. And that is a quite hard thing to do. And I need a map for that. I need a map which tells me what does that genetic variant potentially do to the gene's function. I need that map because I might want to know what other gene could cause the same disease, what similarities are there in these structures. And I might want to have that map in order to help plan ahead. So for example, repeat disorders we just mentioned here, how many repeats? How early will the disorder actually emerge for that patient? And I might want to have that map, most importantly, to find treasure, to be able to define a treatment or cure for my specific patient. So is the current map good enough for the brain? Is it good enough for other tissues? And what I've shown you here is two maps, the map of the world in 500 BC and the map of the world in 2024, even with an epigenetic map on top, which is the weather system if you want. So if you have the map from 500 BC, that map is actually really, really good for the Mediterranean. I mean, if you lived in Italy or you lived in Greece, you'd be great on that map. You're not great on that map if you happen to live in Australia or you happen to live in North America. And so what I would argue is that the map varies in quality. And so we set about trying to define that in a project about 5 years ago, which essentially used short-read RNA sequencing data and asking a really simple question, which is if you map short-read RNA sequencing data to the genome, does it actually map to the exons, or do you have regions which are expressed genomic regions or transcribed genomic regions which are not actually within exons that we know of at the moment? And if you order tissues by the amount of intergenic transcription or intronic transcription, then what actually happens, I don't know if I've got the pointer here, oh yeah. What actually happens is that you see that the tissues on this x-axis, all of these yellow tissues are brain tissues. So we actually have all this expressed intergenic transcription going on, which we haven't really characterized. So we've not got a very good map for the brain. We've got a pretty good map for the heart, it's over there, and we have a better map for the blood. So actually our map is a little bit like that 500 BC map. It's good in some places, bad in others. And we can build better maps now. We can build better transcriptomic maps because actually, as was mentioned before, technology has changed. Technology often drives these discoveries. And we have now 2 different long-read sequencing technologies available to us, Pacific Biosciences and actually Oxford Nanopore, which are readily available and can actually have long-read RNA protocols for them. So if we want to build better maps and we want to navigate to new places, I'm going to tell you about a few stories. One of them is around APP. So here, what we're doing is mapping to a new place in what I would call an old gene. So this gene actually, if we go from what Kathy said at the beginning, that the idea of the Milner is to have human-based drug discovery, it doesn't really get much more human-based than a letter in 1986, actually, which was from Carol Jennings, who is the first, her family was the first family identified to have APP mutations. And she actually responded to an advert from John Hardy, asking for families who think that they might have a genetic cause for their Alzheimer's disease. So she responded, and actually 5 years later, his team found the gene, and that gene was amyloid precursor protein. She unfortunately did not benefit from that. She died in 2024, but she did, and her family has lived to see the development of lecanemab, a drug targeted based on that discovery, in effect. So APP, what do we know about APP? Well, we know that APP is actually crucial. The way APP is cleaved, this protein is cleaved in two places by the beta secretase and the gamma secretase. So you've got these two cleavage positions, which are crucial to generating these A-beta peptides or A-beta species, which are then really the constituent parts of these pathogenic plaques that are pathognomonic for Alzheimer's disease and are absolutely diagnostic for it. So these actual amyloid plaques contain large quantities of A-beta peptides, and those A-beta peptides come from processing of the amyloid precursor protein. So, okay, what happens if we do targeted long-read RNA sequencing of APP? Well, you would imagine that we should find nothing at all. We should find all the transcripts that we know should be there already. And so we actually applied long-read RNA sequencing, targeting to APP particularly. That protocol is online, and we made very few assumptions about what the APP structures would be. So we actually designed lockdown probes that actually went across the introns and the exons of APP, and also the flanking 5' and 3' regions. And from that, what we did was we were able to pull down long-read RNA sequencing transcripts of APP and identify that as well as seeing the coding known transcript structures, complete matches or matches with alternative 3' and 5' ends, we also saw lots of transcripts of APP across different cell systems and in human brain that were actually not known coding. These are actually, would be presumed to encode novel proteins potentially. And we identified those quite commonly. And you might argue, well, okay, that's fine if you identify them, but do they actually contribute much to the total transcription from the locus? And that's what you're seeing here, which is in actual fact they do. They contribute in the cell systems to around 25% and higher in the brain. So does that actually matter? It could do, right? So if we have a look at the structures of APP's open reading frames, those that are in annotation and those that are not in annotation, I want to draw your attention to this novel protein coding sequence of 100 amino acids. And actually, that's quite an important protein coding sequence. And the reason why it's so important, if I can move on to the next slide, is because in actual fact, it's as if you've already cleaved the transcript. So this particular transcript structure actually, in fact, encodes what you might call the C100 fragment. It's effectively as if beta secretase has already operated on APP and added an additional methionine. And so actually that transcript structure is really rather important. It means that you don't need beta secretase activity, and it might give us some understanding of why base 1 inhibition has not been a very effective strategy for AD. So of course, if you don't need the beta secretase, you're already at a C100 structure, you're already at a structure that's perfect for the gamma secretase activity and for generating A-beta. Again, does it matter? Does it happen enough to make a difference to anyone? Also, that hasn't come out terribly well, but the idea is that this APP total structure would relate to these 770 amino acids, 751 and 650, my eyesight's terrible, 695 amino acid sequence. And the one I want to keep your attention to, oh, it's not working, is the C100 at the top. And that C100 is actually at percentage points, so it's not a highly expressed transcript in either cells or tissues. It's of the order of 2% or 4%. But actually, the amount of A-beta that's generated is 2 to 4%. So in actual fact, it sits quite nicely in terms of the quantity which would be sufficient to contribute to A-beta plaque formation. So actually, that's very well that we can find the transcript structures, but can we actually find the protein? And you can see that there's an intrinsic problem with trying to find this protein, if it actually exists, if that C100 transcript is actually translated, in that it only differs from what you expect to find by one amino acid, and that's a methionine. So in actual fact, what you need is particular specialist mass spectrometry, which in fact we collaborated with Henry Zetterberg to enable to get. And what we identified was that indeed, when you take human CSF, you can indeed find these A-beta structures that start at -1, that effectively have that methionine present. And what's demonstrated here is you've got a -1 to 15, a -1 to 15, which is an oxidized at the methionine, and a -1 to 16. And so we actually do know that this is generated in human tissue, in human CSF samples. Okay, so what does that mean? That means that we should be able to generate A-beta peptides without actually having full-length APP. And so we tried to actually generate frame shift mutations, which meant that you drop the quantity of total APP, and that's what you're seeing on APP RNA expression at the bottom there. I'm sorry that I don't think, oh yes, got it. You have to press harder. So you actually, in these knockouts, what you do is you do indeed drop APP total length, but you don't necessarily impact very much on APP C100. As you would expect, that means that you don't any longer recognize full-length APP, particularly from the terminal end. And that in actual fact, though, the key question is can you still generate A-beta 40 fragments in this situation? And the answer is yes. So even when you've got heterozygous and wild-type lines, despite the fact you've disrupted full-length APP, you have not disrupted generation of that fragment. So here, I'd say that building a better map has actually allowed us to navigate to a new place in an old gene, and that new place may be a new amyloidogenic model. Okay, so I've also wanted to tell you about building maps in order to actually make plans. Why do you want to make a plan? Well, you want to make a plan because when you see a patient, you want to know what's going to happen to them. You want to know what state in the disease process they're in. And so here, what we looked at was PD progression. And PD is, again, Parkinson's disease is an old disease. It's a disease that's been described for hundreds of years. And in a way, over time, we've understood that it's a disease which actually really begins far before the point in time that a patient sees their doctor with a tremor or stiffness. It starts way before that. And it will eventually, for some patients, go all the way to late-stage form of Parkinson's disease with dementia. And we wanted to know, really, whether the dementia component of this is actually just more of the same process that started the Parkinson's disease, or was actually, in fact, a different kind of process? Is it more of the same or something different? And so what we did was look at actually individuals which were control individuals, Parkinson's and Parkinson's disease dementia brain tissue. So this is post-mortem brain tissue, analyzed with single nuclear RNA-seq, and we analyzed 2 brain regions, but I'm going to focus on the anterior cingulate gyrus. So there's some sort of demographic details about those, and we had the benefit of having quantitative immunopathology or pathological measurements on these samples. So, but we wanted to move beyond just differential gene expression here, because you might argue, look, differential gene expression is really rather crude, and what we wanted to understand was actually whether we could build a better map, a map that took account of transcript structures, and would that enable us to actually understand the stages of disease more accurately and more carefully than just a gene-level type map. And so to do that, we actually took advantage of the 3' single nuclear RNA sequencing nature of the work we'd done, and actually used a new tool, Scooter, which effectively allows you to quantify transcripts by making use of the fact that you're actually quantifying the 3' end. So what you can do is ask yourself whether actually, in fact, I have a proxy measure for the transcript levels of expression. So in actual fact, many genes have more than one distinct 3' UTR. And what you can imagine is that those 3' UTRs are attached to different open reading frames. Oh, I'm still struggling with this thing. And so actually, you can see that, in fact, depending on the UTR that you're using, you're actually potentially using a different open reading frame. And now, what this enables you to do is actually integrate 2 types of data. Long-read RNA sequencing data that we applied to the samples at the bulk level in order to find 3' UTRs very accurately. They are poorly annotated in human brain, so this was actually quite an important step. And we combine that data with single nuclear RNA sequencing data, and to allow us to align our single nuclear RNA sequencing reads to UTRs, enabling us to do 3' UTR counts by cell type and analyze the actual transcript usage per cell type. So what does that show us? It actually gives us quite an interesting picture. In actual fact, it highlights to us the key changes that are occurring in excitatory neurons, specifically in Parkinson's disease with dementia. So I'll try and take you through this plot. So actually what we're doing is doing two major comparisons, and on the x-axis you're seeing all the different kinds of cell types that we're analyzing. So there are multiple different excitatory neuronal subtypes, multiple different types of inhibitory neurons that we've isolated from the single nuclear RNA sequencing data. We're then comparing transcript usage when we're looking at control samples versus Parkinson's samples, or control samples versus individuals with Parkinson's disease with dementia. And what we're actually asking ourselves is how many genes actually have changes in transcript use, and clocking that up on that top row at the top, which actually has columns for excitatory neurons only, really, and only when you're comparing control to Parkinson's disease with dementia. And in actual fact, I don't think you guys probably can see these labels, but which genes are there that are actually changing transcript use? And actually, you can see that there's APP, pretty important, given actually that as you progress to dementia, there's an increased risk of amyloid plaques in these patients. Synuclein itself, SNCA, is also changing transcript use in the state, specifically in the excitatory neurons. So this gives us a very different picture of the disease than differential gene expression does, which is dominated by effects on the glia. We also asked ourselves whether this was driven by alpha-synuclein or beta-amyloid, and here we benefited from the fact that we had quantitative neuropathology, and so we were able to build into our modeling the alpha-synuclein load or the amyloid load. And what we identified here was actually correcting for the alpha-synuclein load, in fact, obliterated our signals, and that's what you're seeing actually in that top row, there's control versus PD, alpha-synuclein pathology corrected. But if you're correcting for the beta-amyloid, far from actually reducing the number of signals we're seeing, they're actually being increased in terms of actually the number of genes identified and the strength of those p-values, including for APP. So it gives us the impression that actually this is being driven by alpha-synuclein pathology, not by amyloid. So focusing on APP transcript use, what we find is that looking at it, we're seeing that RORB, these are particular excitatory neuron subtypes of layer 4 of the cortex. Those actually drop in their quantity with PD, but not particularly with any further with PDD. And at the G level, APP is not changing level of expression in this neuronal subtype, but actually at a transcript level, some transcripts are being less used and some transcripts are being more used in the state PDD. So just to kind of reiterate the point here that actually there's a need to build a better map here. You're building a better map in order to stage the disease correctly, in order to be able to identify where in this process the patient really is. And for that, you need a detailed transcriptomic map that delves into that and actually describes the features much more carefully. And I think what it's demonstrating is that actually causation and progression are likely to be distinct in Parkinson's disease, that actually there's distinct molecular processes and that actually they may require distinct treatments. And this is something we've already known for a long time for cardiovascular disease. It's not like you try and treat an acute coronary event by actually defibrillating somebody because they're in a rhythm disorder. And it's not as if you try and give them a statin and hope that actually when they're having a heart attack, that's going to particularly help them. So for a long time, we've known that we've got to give treatments that are appropriate for that disease state, but then that means that we need to actually have a map of the disease process. And I think that is where the transcriptomic map matters. So I'm going to go to one last story, which is about, in a way, the finding gold. So here, what we're looking at is SNCA transcripts in dopaminergic neurons. So again, this is focused on Parkinson's disease. And actually, synuclein and the gene that encodes it, SNCA, has actually been in the literature for Parkinson's disease for a long time. In actual fact, if you think back to 1912, Lewy bodies were identified. They were found to be specific to Parkinson's disease and common, actually, in the substantia nigra, particularly dopaminergic neurons. We then identify that actually the constituent part of Lewy bodies or major constituent is the synuclein protein itself, and that in actual fact, mutations in the synuclein gene give rise to early onset Parkinson's disease, duplications of this gene, triplications of this gene give rise to early onset Parkinson's disease, and actually even common variation around this gene increases your risk of Parkinson's disease. So this gene, in terms of the dosage of this gene, the type of this gene, its actual amino acid sequence, all impact on the ability of alpha-synuclein to aggregate and to cause Parkinson's disease. So again, what we did was use targeted long-read RNA sequencing, this time applying that to iPSC-derived dopaminergic neurons, and we studied control dopaminergic neurons, those with an A53T mutation and those with a synuclein triplication. And these were highly well characterized in Sonia Gandhi's lab at the Crick. And again, you might say, and I remember Sonia saying, I don't think we're going to find anything, we're just going to find one open reading frame of synuclein. In actual fact, again, that is not what we found. We found novel coding structures for synuclein, and most maybe disturbingly for cell biologists, we found that those novel coding transcripts were actually relatively commonly used. So here on the second plot, I'm sorry for the pointer problems, where you see transcripts ranked by expression, we're ordering the transcripts we detect based on actually how commonly used they are. And the astute amongst you will notice that that y-axis only goes to 30, i.e. the most commonly used transcript that is in these pure cell types of dopaminergic neurons at 85%, 90% purity is actually only being used at 30% of all the transcripts being made. And the fifth most common transcript is actually a pinky one, which is actually, a purple one, which is a novel coding transcript. So, this is not what we expected to find. We were very robust and severe with our quality control here. So this has generated 100,000 full-length reads per sample. A transcript had to be detected in all samples for us to consider it to be there, and it had to have a minimum usage of 0.3%. And that means that you have to have 300 full-length reads supporting that transcript structure. Frankly, when you look at your normal annotation, a transcript level, TSL level 5 gene or the main select transcript structure usually doesn't have that much data behind it. So does it matter though? I think it does matter because it could have implications for two things: the aggregation of synuclein, and it also could have implications for how you might actually want to target synuclein. And in actual fact, for those of you who might not be in the field, there is actually a phase, synuclein ASOs are in trial, doing exactly that, trying to target the messenger RNA to lower the levels of synuclein. So, the reason why aggregation might matter is because actually the alpha-synuclein monomer structure, if you've changed it, if you've made it more hydrophobic, could contribute to the ability for it to form fibrils, to aggregate more generally, and ultimately to form a Lewy body. You might argue that the RNA structure itself is also important to you because if you're designing ASOs against that structure, well, you wouldn't design a drug without knowing the protein structure that you're actually trying to design against. So why would you design for an RNA without knowing the RNA structure? So actually these novel transcripts of SNCA that encode novel proteins, we looked for them in two different ways. I'm not going to go through them in detail because I'm going to focus more on the ASO component of this. But we created a pipeline where we actually didn't generate our own mass spec data, but simply searched public data. Actually, the software to be able to do this is now just gone on bioRxiv about 2 days ago, and we call it transcript to protein. Effectively, what you're doing is calling the transcript structure, then calling the open reading frame from that, and then searching for peptide sequences that would be unique to that structure that are long enough, and effectively going into the mass spec data sets that are in the public domain to ask whether you can see those peptide structures with high confidence. So we did that for SNCA, and we actually found that indeed we could detect SNCA novel peptides using this approach in the case of at least 2 novel peptides, where there were unique amino acid sequences. And those have a predicted length of 113, 118 amino acids. And interestingly, those proteins would be predicted to have a much lower solubility, something that would be expected to impact on their aggregation. We also actually did pull downs using antibodies to select for synuclein from human postmortem brain tissue, both anterior cingulate gyrus and the substantia nigra, and again demonstrated that we had mass spectrometry peaks that would only be explained by these novel structures. But let's have also a look at leveraging 3' UTR variability to design ASOs. So actually what we're showing here in this first plot is a variable 3' UTR lengths. So some of those UTRs have very different lengths than others, as you can see here. So this is literally just plotting UTRs that we detected for synuclein. And actually those different UTRs are actually attached to transcripts which have a different relative expression or use. And so you can see from that that actually this is pretty important data if you want to design an ASO. Because you might argue that depending on where you put your ASO, you'd be targeting a different range of transcripts. And indeed, that's what we showed here, is we actually did ASO designs that were across that whole 3' UTR space. We then looked at the relative impact of those different ASOs, numbered 1 to 9, on the effects on lowering SNCA transcript levels. So then actually from this, we can see that in fact ASO1 is our most useful ASO. If we apply that ASO to dopaminergic neurons that carry the triplication and we compare it to scrambled ASO, what we can actually see is that in fact we do indeed lower synuclein protein. This reduces synuclein aggregation, shown in the second column of data, and actually this reduces neuronal death. And so actually, we think that efficient ASO design could be done through better understanding the transcript structure, and that these would be safer ASOs too, with a reduced side effect profile potentially. Furthermore, what we've realized is that actually different SNCA transcripts are expressed by different cell types. So if we look at astrocytes and microglia, they do express synuclein. They express actually different transcript structures with different 3' UTRs. They don't necessarily vary so much in terms of the amino acid sequences. And actually, depending on the astrocytes, whether you treat them with LPS, or the microglia, whether you treat them with something inflammatory or not, you can actually predict that you would expect to get different levels of suppression of SNCA that would be dependent on cell type and cell state. And so that gives you an opportunity to treat in a highly directed manner if you actually designed against the real structures that are present. And of course, that makes the data that I showed you earlier about the fact that SNCA transcripts or APP transcripts are changing in disease states, you would need to account for that in doing effective ASO design in those situations. So I was just going to finish up by making the point again that I do think that genomics has the potential to transform neurological treatments. I'm a clinician scientist who's trained in clinical genetics, and there's no doubt in my career we've gone all the way from sequencing single hotspot mutations to going to exons, to going to whole gene sequencing, to whole genome sequencing. So I have no doubt that we're going to move in time to long-read sequencing, and I have no doubt that that data is going to become effective for treating neurological diseases. But I think in order to do that properly and do it in a directed manner, we actually have to be able to interpret the data we have there, and we need a better map not just of the genes, but of the transcripts they generate. So that leaves me just to thank the people who did a lot of the work here, and that's most importantly, actually, the patients who donated samples and brain tissue. So many of these genes are human lineage specific in terms of their sequence properties, and I think it is key that we have to study these diseases in actual human tissue, and that depends on patients being generous with their donations. I also wanted to thank members of my lab, both past and present, Aztecs Pharmaceuticals, who contributed to some of the software that we used, the Gandhi lab, who we collaborated with for the dopaminergic neuron work, also Charlie Arbour and Selena's lab at UCL, who we collaborated with on the APP work, and lots of members of UCL like John Hardy, Henry Holden, Nick Wood, who contributed to the single nuclear RNA-seq data generation and so on. So, and of course, our funders. So thanks very much. No, I was pressing really hard and struggling.
We have time for probably one, maybe two questions. Let's see.
Sorry, I went to time, didn't I, to the full 30 minutes? So.
It's all right. You were having problems with the pointer. It's very funny. Questions? Anybody? There's one there, yeah.
Thank you. Very exciting work. I'm just alluding to your last bit of your talk where you talk about the 3' UTR and the cell specificity. Just curious because maybe I missed the connection, but you showed the expression of the ASOs. But can you speak to what the knockdown effects are based on knocking down those particular ASO? Do they also translate in the same therapeutic window as you would expect them to?
I mean, I think that's obviously key. I mean, I guess what we can see at the moment is that in actual fact, these are predictions. So what you were seeing there was what the relative usage of the different transcripts is, and therefore what the expected impact of an ASO design is in one position or another. At the moment, that's exactly the experiments we're running, which is to try and test out whether those predictions follow through in terms of what really happens. Where we do know that is where we did the ASOs on the dopaminergic neurons themselves, where we know that different ASO positions do indeed very correctly predict the efficacy of the ASO in dropping down SNCA. But actually, of course, what we want to do is know whether if I took a mixed culture of cells and I applied a particular ASO that was designed for one UCR versus another, would I only treat one cell type? You know, that's the key experiment, right? So that's basically what we're doing at the moment.
Okay, I think, there's one more. Yes, one, do you see over there? Go ahead.
Hello, thank you so much for the wonderful talk. I had a question related to the astrocytes slide that you were showing. Well, recently we have understood more about tau, even though it's not a protein that's expressed in astrocytes, but already the transcript versions of it, the mRNA versions of it, for instance, can, aggravate the disease. But SNCA or APP are also produced as proteins in astrocytes. So do you think that has a similar take to, for instance, tau-related, aggravation in the disease? Or do you think already the transcript forms of it can cause or has involvement? Have you looked into those?
So, I mean, we do have a separate project on tau because tau, of course, has splicing mutations. Usage of exon 10 is directing whether it's 3-repeat or 4-repeat tau, and tau also is known to actually mature in its splicing. So in actual fact, unless you age your cells out to almost a year, you don't actually get mature splicing of tau. So I mean, I think it's certainly an area where we imagine that. I can't comment on tau expression. We did not find changes. I actually thought that we would find changes in tau transcript use in PD, actually in PDD. That's what I thought was more likely than the changes in transcript use of APP or synuclein, in fact, because the MAPT is also a key disease locus for PD with the H1H2 haplotype. But I don't have specific data on astrocytes and MAPT at the moment, in all fairness. So I don't know yet, is the short answer.
Looking more into the glial cells now, which I think is already...
Yeah, I mean, actually, as much as 5-6 years ago, we published a paper which said moving beyond neurons for Parkinson's disease. So I definitely think we should stop just spending our time on neurons. Yeah.
Okay, let's thank Mina one more time.